Chapter 15, Rhabdoviruses. Minus strand RNA. So this is the, not messenger RNA, but the template for messenger RNA. They are enveloped. Smaller genome, bigger than parvoviruses, lower than herpes viruses. 11 to 15 kb, that's enough to code for several polypeptides, as we'll see. Derived from the Greek word for rod, so because they are in fact rod-shaped. They look to me like bullets. That to somebody at some point naming them was a rod shape. They have a wide range of hosts. Here you can see all sorts of furry animals, raccoons, dogs, cats, bats, mongooses. I just been bitten by a mongoose in rural South Africa. But anyway, the host for the disease. As we've already talked about this with respect to rabies, generally a puncture wound through skin gets into the neurons. Neurons are nice long cells, so infection goes from cell to cell, and of course that causes the neuropathologic effects. We showed you that awful video of the guy dying of rabies, occurring due to the infection of the neuron, but propagation of the virus requires it to eventually get to the salivary glands, where the virus is produced in saliva and goes to the next host. Almost always fatal. Oh, sorry, I've moved into rabies, so I guess there are non-fatal rhabdoviruses. So we have a viral envelope, so the overall rod-shaped viral envelope, several polypeptides encoded in the genome. We used to have an ironclad rule in graduate school that proteins interacted with each other in alphabetical order. Because in my field, genes are named for where they are in the genome. So A, B, C. I was just hoping it was here, but this is not alphabetical order, so never mind. 5 genes encoding proteins for nucleoprotein. Encodes N, nucleoprotein, so it's an RNA binding protein. P and L, this is a rather easy nomenclature. P is a phosphoprotein. And L is a large protein. P and L, LP3 stoichiometry, have the RNA polymerase activity, among others. N, P, and L are all nucleic acid associated, which in this case is RNA. So RNA associated proteins, N, P, and L. We have M for matrix protein. You see here L, the large protein, N, nucleoprotein, P, phosphoprotein. We have an LP3, so that's going to be our polymerase, but it's found in the nucleic acid in the virus itself. N not only coats the RNA, which has a protective function in the cytoplasm, I'm sure, but also organizes the genome with respect to the matrix protein, which is inside the viral envelope. When released from the matrix within the virus, it turns down host gene expression. It's the way the virus has of taking over the host in order to make viral proteins. And then we have G for glycoprotein, which is, of course, in the spikes that govern attachment. So one molecule minus strand RNA, so upon infection, mRNA can be made directly, as we'll see momentarily. Look at me, I'm slowing down. So replication cycle, once again we have host protein spike interactions, more than one, so there's your chained equilibria leading to high affinity attachment that leads to endocytosis mediated by clathrin. There's also an additional component of actin becoming involved after the clathrin-coated pit forms. So two components of the cytoskeleton are involved. We have dissociation of clathrin from the endosome followed by escape. Although in contrast to other viruses, the particle doesn't escape, but the viral membrane fuses with the endosome, resulting in escape of the genome, but retention of the glycoproteins and some of the matrix proteins. So you see the dots and the spikes. So the matrix protein, in addition to remaining behind with the empty viral envelope, goes to the nucleus, turns down host cell expression. The nucleic acid remains in the cytoplasm, so we have the minus strand and RNA-dependent RNA polymerase. LP3 is an RNA-dependent RNA polymerase, so immediately messenger RNA can be made and translation can occur to make viral proteins. Virus regulates the amount of protein it needs. It needs fewer glycoproteins than nucleoproteins. It needs fewer large proteins than phosphoproteins. It's a 1 to 3. And so what we have is transcription of the mRNA, transcription of mRNAs drops off as we go down the genome. We have a binding event, and then it can fall off at various points. So the arrangement of the genes within the genome relates to the degree of expression of the production of the viral proteins. More N than L. And that's a function of the structure of the genome.
Can you say it one more time that it relates the arrangement of the genes in the genome relates to?
Yeah, so transcription occurs in a 5' to 3' direction. So this is the, 5' of the minus strand, 3' of the minus strand. So if I'm the mRNA, I set down there's a leader sequence here. So it's starting transcription at that leader sequence, and as transcription occurs stochastically due to binding, right? We're talking about protein DNA forming and then dissociating. That's, we can talk KDs and whatnot, but transcription occurs, and just stochastically, which is a fancy word for meaning randomly, the RNA polymerase will dissociate from the transcript, so that for every transcription event that starts, you have a much higher probability of making a nucleoprotein, sorry, I'm blocking you, than you do of making a nucleoprotein and a phosphoprotein. The chance of making both of those is even higher than M, and so forth. So that transcription events starting from here, very frequently are going to make nucleoprotein, less frequently are going to make large protein, which works out. That's more efficient for the virus, because to make a new virion, look at that, you need a whole bunch of nucleoprotein. You only need a couple of glycoproteins, one large protein. So being down at this end of the genome, it's right here, is where you want your low copy number proteins with respect to the completed virus. So the arrangement of this helps the virus be more efficient in producing progeny virions.
So what, did you go back?
That was very fast, wasn't it? I was just saying, oh, I said that fast. I should slow down.
You go back a couple slides. Get that one right there. So you said that the N particles, or the N proteins, is replaced.
So the matrix protein has two functions as far as we're concerned here. One of which is structural. Let me go back to this. Right, so it forms the matrix. So that's my Cambridge impression. Forms the matrix inside the envelope. So there is a structure, the virion function, to organize. You see, it's showing a nucleoprotein matrix protein interaction. So there's a binding event there, which keeps the genome properly arranged in the virus. So that's a structural virus protein. It's multifunctional in that upon release, it's transported to the nucleus, so somewhere within the sequence is a nuclear localization signal. It gets in here and shuts down expression of host genes and turns them down. The upshot of that is the RNA that's made here is going to be translated more often because we have less host RNA being produced.
Okay, so on that last little part right there, I can still see
This?
No, up top. Yeah. Are those just, do they stay there?
Yes. So they stay there, of course, because they're bound
Right.
to the what was the membrane envelope,
Okay,
and they're
so
not
just-
dissociated.
Okay, okay.
There are other, so, but you see in this rendering, that's a solid color.
Right.
This means more, less. Some of that more that's not in the less is here in the nucleus.
Okay. I was just making sure that's what was actually happening.
Yes, it's not a switch. It's not like all of the matrix protein goes to the nucleus. Multifunctional, so some of it, this is because of random binding events. Tight binding is not going to dissociate as often as weak binding. And so it's appropriate, I mean, it's an evolutionarily designed balance, right? If it's stuck too much here, it would be turned down host expression. If it came off too easily, it'd be a crappy structural protein. Yes, awesome.
Does that get degraded, or does that, the new virions get packed up in?
I don't know. My guess is it gets degraded. Because what we would have here is these proteins would be external to the new endosome. So the biophysical, well, there's going to be some viral effects on the structure here, but you've lost your endosomal integrity, so endosomes have a lower pH. When this fusion event occurs, this is not what the inside was there. So my guess is proteases take care of it eventually, and it's new virus that has to form.
Would the nucleoprotein and the matrix interactions be like a scaffolding protein?
Yeah, you could think of it that way. But it's not a scaffolding protein the way scaffolding protein in, I guess the virus is scaffolding. There are other, the one we've talked about, there are lots of viruses in scaffolding. It's not scaffolding protein like we've seen before, insofar as it remains in the mature virion. So the scaffolding protein, herpes? Yes, herpes. I got it right. What a good matching question. Is degraded before the mature capsid is formed. Right. Small genome, so we only have a couple of components we have to worry about. So the N, P, and M proteins are all translated in the cytoplasm. As is the L. So 1, 2, 3, 4, the 4 structural proteins that are soluble are translated in the cytoplasm. G is a glycoprotein. Remember, glycolysis occurs within the endoplasmic reticulum. So it's synthesized at the rough endoplasmic reticulum, like all membrane proteins in eukaryotes. And as it's translated and integrated into the membrane, we have glycolysis, transport from the endoplasmic reticulum to the Golgi. And at the point where we get to the Golgi, we have transport to the cell surface and glycoproteins arranging with matrix proteins within the cell surface, recruit matrix in protein, and then a copy of the RNA. We'll talk about replication of the nucleic acid in a moment. For assembly of the full virus, the viral envelope is part of what was the cell membrane, and we have budding of the virus off, so there's an electron micrograph and new viral particles getting ready to bud off. should be enough. So remember, we're a minus-stranded RNA virus. The replication of the nucleic acid is not something that the host protein is going to have machinery to do, but rather we have our RNA-dependent RNA polymerase, which can do a couple of things. It can synthesize the messenger RNA, which can then be translated into, why is that pointing the other way? The plus strand RNA can be translated. It can also serve as template for synthesis by LP3 of new minus strand. This is wrong because the plus would be translated, but the minus serves as the progeny genomes. So the same LP3 does both plus and minus strands. It's the minus that gets packaged. Why would it be the minus that gets packaged and not the plus? Well, but viruses are stupid. How are they going to know not to package the plus? I'm sorry? So it's used as mRNA, would be bound by ribosomes. I don't really know what the answer is, but nucleoproteins can specifically bind to the minus strand. That's almost surely what the answer is. The specificity of protein-DNA interaction results that the minus, actually we see plus strand RNA bound there by nucleoprotein as well. So much for that. I still think it's right. That's all I want to say about rhabdoviruses. And I want to spend a lot of time on influenza because it's all interesting. I will start with an animation I found yesterday. There's an interesting story behind this. So this is going to be an animation of influenza viral replication cycle. It's done by a company here in the Atlanta area that was spun off of Emory that has a novel drug target identification strategy. And the link for the YouTube video is in the PowerPoint, written in. So you can watch it at your leisure, and I think it's better than my explanation of who's by replication cycle. I'll go through that as well. But I have an interesting little aside after. Hit pause. I'm going to move out there. Pause.
Now, no, pause.
All right, that's close enough. There we go.
A virus is a small infectious particle that hijacks the machinery and metabolism of our cells to grow and reproduce. Each viral particle, or virion, consists of nucleic acid, either DNA or RNA, in single or double-stranded form, enclosed by a capsid. In most viruses, the nucleocapsid is enclosed by an outer envelope. Influenza type A, an enveloped single-stranded RNA virus, has been widely studied and serves here to illustrate the life cycle of one common virus. Scientists believe that influenza virus is spread from person to person through exposure to large respiratory droplets, direct contact, or airborne dispersal. Infection takes place mainly in the respiratory tract. Infection begins with attachment of virus proteins to a receptor on the surface of the host cell. The virus is then taken in the cell by receptor-mediated endocytosis. As the virus enters the cell, The virus is internalized in a membrane-bound capture vesicle that carries the viral core. The vesicle is transported on microtubules inside the cell by host proteins called kinesins. During transport, the membrane of the vesicle fuses with the membrane of the virus, and the capsid undergoes uncoating. The viral core RNA and proteins are then released into the cytoplasm, where they are guided by host proteins to the nucleus of the host cell. At the nuclear membrane, the viral core uses host protein channels to enter. Inside the nucleus, cell machinery is utilized by the virus to replicate the viral genome and make messenger RNA, mRNA. Some viral mRNA exits the nucleus to exploit cellular ribosomes to direct synthesis of viral proteins. Viral proteins go back to the nucleus to associate with viral RNA. These nucleoproteins again leave the nucleus and use cellular processes to travel to the cell surface. Viral surface proteins are made and processed in the cytoplasm and also travel to the cell surface, where they combine with encapsulated nucleoproteins to form progeny viruses which depart from the cell by budding. The virion now goes on to infect other cells. Due to the limited number of viral proteins and nucleic acids that can be targeted, the development of effective antiviral agents is especially challenging. Drugs currently used to treat influenza act by inhibiting viral proteins involved in budding and uncoating. However, these antiviral drugs are of limited effectiveness and, like antiviral drugs used to treat HIV and other viruses, are prone to development of viral resistance through mutation to avoid the interaction with the drug. At present, vaccines to stimulate the production of antibodies against the virus and stimulate host-cellular responses to ingest the virus remain the best strategy for controlling certain viral diseases, but they are of limited effectiveness against rapidly mutating viruses and are ineffective against viruses that cause the common cold and AIDS. Scientists at Xyris are working on new and better ways to prevent and manage viral infection by developing a platform of host cell-based targets to inhibit viral replication. To find these targets, we are using a patented technology called the gene trap. Xyris scientists transfect each cell in a colony of identical cells with a retrovirus, a small particle that causes a random insertion into the cell's DNA. The insertion randomly disrupts the function of a single gene in a cell into which it is inserted. Collectively, these cells create a unique library of cells, each having a different single gene disrupted. Importantly, cells that require the disrupted gene to survive will die and are not identified by the gene trap. Thus, there is a selection against genes that are essential for cell survival and likely toxic if targeted with drugs. Only the more promising drug targets are identified. Xyris scientists then infect every remaining cell with a virus that normally kills all cells and identify those cells that survive the infection. Since all cells would normally be killed by the virus, the surviving cells must have survived due to a gene disruption of a human host gene product that the virus must use or hijack to replicate. These specifically identified genes and their RNA and proteins, when turned off, prevent a particular virus from replicating. The Xyris gene trap represents a cost-effective and efficient platform for the discovery of targets and drugs to block viral infection. Using this technique, we have efficiently discovered hundreds of human cellular targets essential for viral replication. Using these cellular targets, we then develop drugs that selectively block the function or production of the target gene products. By inhibiting cellular proteins critical to the virus, we stop the virus in its tracks and prevent it from growing and spreading. Based on in vitro data, by blocking cellular targets, we can prevent any type of virus from replicating. Significantly, many of our targets can be utilized to block several different types of viruses. For example, based on in vitro data, blocking a single Xyris gene target can block Ebola, influenza, and HIV. Thus, using our platform, effective broad-spectrum antiviral drugs, which we call Xyris antivirotics, are now possible. In addition to viruses, we are also currently applying Xyris technology to target multi-drug resistant bacteria. as well as bacteria that are listed as bioterror threats by the CDC and the NIH. The Xyrus approach could provide the keys to preventing and treating infectious diseases and future pandemics, whether made by nature or mankind.
So the preceding did not constitute an endorsement of Xyrus or its technology, but it wouldn't matter if it did. I stumbled across this yesterday when I was prepping for class. I wonder what Xyrus is all about. So I went to, because I like the animation and I saw the strategy. It seemed like a great idea, right? Because it's a very real point that antivirals directed at viruses, even when they're good, are of limited utility because the viruses will mutate very rapidly, but host targets that are needed for replication, if you can inhibit them without great toxicity. You saw their screen at least, well, it's toxic, it will kill the cells, is a really neat idea. So I went and typed in Xyrus.net, which is their website, and I got this, which is a law firm claiming that they have seized control of the domain for alleged counterfeit Uggs products. So, you know those shoes?
Oh, wow.
I suppose what they had was a nucleic acid sequence, the Uggs that they own. I don't really know. The company hasn't issued a press release in like 3 years, so right after this, appears right after this animation got posted that they went belly up. They were a startup spun out of a lab at Emory. I assume what means is, that's really awesome, but it didn't work. But it's almost impossible to find. If the interesting aside was, and I forgot to plug it in class, our speaker at the seminar on Friday came from the lab that spun the company off. In his talk, was the apatoisomer thing that I didn't know what it was. It turns out that's an antivirus thing. That he was looking at crystal structures and developing inhibitors for broad spectrum antivirals. So it was something if I'd known 72 hours earlier, I might have had an even more interesting story for you here. But you know, the idea when you start your biotech company is to start it, get the money as soon as you can, and get out, because it's almost surely going to fail. Not that that's a bad thing, right? If 95% of companies fail and 5 of them make good products, and they've probably been worth the investment. Let me also take a moment to get a plug for the book. How many people are reading Influenza? Oh, good. So you'll note on this, so flu virus, the history of this is, to me, is fascinating. At the end of the semester, we'll come back and do a class on the book after everyone's read it. If you hadn't started, it's a really fast read. It's not very thick at all. Great book, and then you have the opportunity to not only just read it for fun and enjoyment, but also to do a book club discussion for LCA points and turn it into a term paper. And I'll have more details forthcoming on that soon-ish. Anyway, that's that. So influenza viruses, minus single-strand RNA. Envelope. Anybody know why they're named influenza? No, but good guess. So anybody have a guess as language of origin? Oh, come on, make it get Greek! Right, that's got a good chance of being right. Wrong. Wrong, but good guess. Italian. Which is really weird. Named after, at least it's speculated, the influenza, the influence of the cold. I don't remember what the Italian is for cold. But the disease symptoms were first described and related to cold weather. So in Italian for influence. we'll come back to cold in a bit. Unlike other minus-stranded RNA viruses, it replicates in the nucleus. So we'll see a slightly different replication, not slightly different. Care that you know that it's Orthomyxoviridae. There are 3 genera or genuses. Influenza, this one's really easy, A, B, and C. The most devastating one, at least from a human health point of view, is influenza A. It is working in the lab, it is spherical. In the wild, it shows spherical and elongated shape. It has a segmented genome. So you saw in the animation, they had the correct number of RNAs, which is 8, because they were talking about A and B. C. They did not, however, in the, just let me fill my soapbox for a moment, cytoplasms are nothing like they're shown in the animation there. They're not a big bag of water like a swimming pool. There's a lot of other moments if you show those in the animation, it's terrible, so we never did. Anyway, 8 RNAs encoding 9 proteins or types of proteins. Labeled here in your figure, we have hemagglutinin and neuraminidase on the envelope surface, the glycoproteins. Hemagglutinin is what binds to the host cell. We'll get to neuraminidase in a minute. One T or 2. What? So one of your entry into the course was what's hemagglutination. It's the ability to make red blood cells clump. So those bind to red blood cells. There's more than one on a viral protein. So if you drop some hemagglutinin into a tube of red blood cells, it will clump up, hence the name. Neuraminidase is a membrane-bound enzyme. It hydrolyzes neuraminic acid, which is the host molecule, also called sialic acid. So hemagglutinin binds to sialic acid. That's the attachment for attachment and entry. And then we have M2 also in the membrane, which is an ion channel. There it is. In the hemagglutinin and neuraminidase are responsible for strains. So different strains of influenza go by different types of hemagglutinin and neuraminidase. So that's the antigenicity of those 2 proteins. And this, yeah, that's right. The antigenicity of the strain is from H, so if you have H5N1, you'll see that in the news, or H1N1. I believe the Spanish flu was an H1N1. That's hemagglutinin type 5, neuraminidase type 1. So there's different mixing and matching in flu strains, not types. So type A will have different strains of that are described by which serotype or antigen type the virus has. And when you next see a news story about the new swine flu that's breaking out in Singapore, they'll tell you what these are, which doesn't mean anything to you except for all of it. The RNA segments or RNA protein complexes are RNA protein complexes. So there's your RNA, it's coded in a protein and then large protein complex. Again, we'll come back to that. Oh, by the way, that alarm that went off during the movie marked the exact halfway point in the course. So you're on the downhill slope. So the replication cycle, like all replication cycles except ones that directly penetrate, starts with attachment and entry. And so mostly, as you saw in the animation, as mentioned in text, it's the respiratory epithelium so that the bad thing about flu viruses, of course, is that they're airborne. Isn't that really gross, thinking you're sitting in a room inhaling droplets of other people's spit? Happening right now. So we have the hemagglutinin, which I'll abbreviate HA neurominic acid binding. So this is a cell surface carbohydrate derivative that's involved in cell-cell contacts. We have HA binding. We have your 8 ribonucleoprotein complexes. We have, once again, endocytosis after attachment. So going from extracellular milieu to endosome brings the proteins into an area of more acidic pH. That causes a conformational change in M2, which opens up the ion channel, allowing further drop in pH. So you see this rendered in the figure by increasing proton concentration. So more protons equals lower pH. That acidic environment causes a conformational change in hemagglutinin. So hemagglutinin is proteolytically cleaved but linked by disulfide between domains. So we have HA1 and HA2. If you look at projections of the tertiary structure, there's a change between uncleaved and cleaved. If you render that as a blob, it looks like this has a head and a tail, head being HA1, tail being HA2. That conformational change in HA leads to, let me come back. So we have M2, ion channel allowing more protons in, so that further drops the pH. Two things happen. That change in hemagglutinin leads to fusion of the endosome and the envelope. This is not escaping the endosome, but rather fusing with it, so we go from 2 membranes to 1. And it allows the ribonucleoproteins to dissociate from M1, which is within the structure of the virus itself, and escape, from whence they are transported to the nucleus. So attachment entry phase ends with the nuclear transport of the ribonucleoproteins. So attachment and entry, first 2 phases of ATGA. Attachment, of course, ends with endocytosis, and then escape of the ribonucleoproteins, which is brought about by these 2 different conformational changes of the ion channel, which leads to fusion, which leads to conformation change in HA fusion, and dissociation of the ribonucleoproteins from the viral body, allow escape of the ribonucleoproteins, which are transported to the nucleus. So at that point, the ribonucleoproteins have entered the nucleus. The replication, so it has, it's a RNA virus, so it has a viral RNA polymerase. So the plus strand is messenger RNA. There is a viral protein called non-structural protein 1, which inhibits cellular mRNA production. So this is akin to the matrix protein of rhabdoviruses, inhibiting their cellular gene expression. There are 10 messenger RNAs from the 8 segments. And here's a reason probably underscoring the need for the minus strand to be in the nucleus, is that the splicing occurs, which gives more variation in the proteins that can be made by the limited genome. So in the genome of 10 messenger RNAs, splicing allows for a wider variety of proteins, but the virus doesn't encode any of its splicing machinery. So messenger RNA synthesis in the nucleus allows for that. So the minus strand actually steals a cap from the virus steals a cap from other messenger mRNAs. Caps plus strand that goes to mRNA with a cap and polyadenylation occurs. Plus strands that go to the template for the genome for packaging is neither adenylated nor capped. These are the possible messenger RNAs, just an example of in genome segments 7 and 8, the splicing that allows for 2 different messenger RNAs from the same genomic template, and then there's another reading frame within genome segment 2 as well, and you just see that say 4 codes for hemagglutinin. Don't care that you know these details. Would care that you, for example, know why influenza has to go into the nucleus. Or why the the replication cycle of influenza goes via the nucleus. So, as in other viruses, we have our mRNAs exported to the nucleus. Exported from the nucleus, so translation occurs inside. Translation's occurring in the cytoplasm. There are proteins that encode the mRNAs back in. You saw in the video translation and then re-importation into the nucleus. The hemagglutinin, M2, and neuraminidase are all glycoproteins, so they are synthesized at the rough endoplasmic reticulum, transported to the cell surface where they collect. The mature minus strand ribonucleoproteins are exported from the nucleus as well. This could be by diffusion and binding, packaged to the nucleus where they are collected and fused or bud off of the host cell. So this is post-Golgi trafficking to the cell of the glycoproteins, packaging of the ribonucleoprotein, budding off, and here's where the neuraminidase comes in that we have a very strong receptor-ligand interaction of high enough affinity, very low KD, that our virus, it's important that the virus is never going to be free. It's never going to dissociate. The equilibrium is shifted almost entirely toward the bound state, such that the neuraminidase, the membrane-bound enzyme hydrolyzes the neuraminic acid, leading to release of the fusion of the progeny virus. This will be important. Well, it is important, and we explain as important later on. So there have something for
So, if you were asked on the test, where does translation occur within this virus, we would say cytoplasm, right?
and rough endoplasmic reticulum.
Okay.
Translation always occurs in the cytoplasm and the rough endoplasmic reticulum. The difference between this virus is RNA and say rhabdoviruses. Rhabdoviruses are weird insofar as any virus can be weirder than other viruses, in that it does its replication in the cytoplasm. So the reason why influenza, new influenzas break out is it has a relatively wide range of hosts for humans, human history. It's we have domesticated many species, plants and animals. Human proximity to animals that can be infected with the virus as well is what leads to emergent strains. So we have influenza strains that primarily infect poultry. They have reservoirs in the wild, so that's a non-domesticated species. And we have transfer among species to domesticated poultry. This is also true of swine. So your swine flus and bird flus, those are influenzas that are making a leap from birds to humans, which is generally shown as somebody living in the developing world, because we in the developed world tend to not be so close to living poultry all the time, or swine flu. Although the 1918 Spanish flu, anybody know where that broke out? Some of you are reading the book, yeah. Kansas. And actually the book goes and finds like 3 people who might have been the first infected with it. So it's by no means exclusive to the developing world. I guess 1918 the U.S. is much less developed than it is now. But this leap from species is where new strains come from, because we get, we don't have the vaccinations and et cetera when new viruses emerge. Oh, why do we have flu season? That's one theory. We're inside. So you guys know there are there are theories as to why there's a flu season. There's nothing about that replication cycle I just showed you that would lead you to think about seasonality of the flu. But as doctors will tell you, they don't see flu in September, they see it in December and January. So, immune system, so there's some speculation that goes to, we're not out in the sun enough, so we don't get enough vitamin D, and that's an immune suppressant, so we're more susceptible to illness in the winter. People are inside in the winter. Kids are in school. Well, it gets colder in the winter, but
Well, it's cold outside and you get the heat on inside. So your body's fluctuating.
oh, I guess additional immunosuppressant. There are a number of other theories as well. Turns out, I just learned this yesterday, they're all wrong. And we've now known for about 7 years why flu occurs in the winter. And the answer comes from the primary literature. The scientists doing experiments and writing them up in a paper, making people, well, they just put it out there and interested people read it. And this was actually covered in the New York Times. It's where I found it, traced my way back to the paper. So in 2007, a paper came out, PLOS Pathogens. So PLOS is Public Library of Science. It's an open access journal, so anybody can read it. The downside to working with flu is, well, there are a lot of infectious diseases that are like this. We don't have a model system. You can't go around doing experimental stuff in humans, you get into trouble. But mice don't get the flu. Or they can be infected, but they don't transmit it. So mice that are perfectly adequate model system for a lot of other diseases, useless for flu. Other organisms like ducks, one, they're probably hard to work with, and two, they're not like humans in many ways. Yes, ma'am?
They couldn't grow like human cells in the media.
So you can do them in, yes, you can do flu tests in vivo, but, sorry, in cell culture, but, you see, you can go into endothelial cell culture, for example, but they're not going, it's not showing you, it's not a model system for transmission of the virus. You could study, and we do, attachment, entry, replication. That replication cycle I showed you was almost surely worked out almost entirely, I would guess, in cell culture.
Random facts. They'll pay you. I just got a letter just a couple weeks ago saying CDC would pay you like $3,000 to infect you with flu and quarantine you. Yeah. I did. I got it just the other day.
Are you going to do it?
No.
What would
Spring
it take?
break.
How many LCA points would that be worth?
Depends on how much money the CDC is going to pay me. But so this actually is an example of this guy. I forget the professor's name. That's the graduate student there, Lowen, who actually did the work. Was reading literature from the 1918 flu. So a paper that was published in the 20s that were observations from an army camp where the flu broke out. I believe it was in New Mexico. I can't quite remember. And the guy had just noted that the guinea pigs in the hospital lab were dying. And they thought it was some food poisoning. The guy goes, oh, guinea pigs. So they decided to test. This was in, I think it's Columbia University in the early 2000s, so 80 years later. Maybe guinea pigs are a decent system. Why nobody thought to try it, I don't know. But the guy went and read a 75, 80-year-old paper and thought, okay, well maybe we can do guinea pigs. And sure enough, they did. So this is the experiment. You say papers are difficult. Here's a place where it's easy. That's a guinea pig. If I ask you this as a figure on the test, be able to interpret this. So they have a change. So this is where cell culture wouldn't work because it's not one organism to another. You could see a budding, you could probably experimentally observe budding of a virus and then its fusion and entry into a cell right next to it, but that's not the way flu is transmitted here. So what they did was set up a cage where there was a flow of air across the larger cage, and then they would put flu-infected guinea pigs in one cage, and downwind they would put a naive, this is the term, naive guinea pig, and then measured, and so you can tell they're infected because they have red eyes. And then look to see whether the exposed guinea pig got the disease. And so the cool part is that this was an animal model for transmission of the flu virus. So there were controls here, they described them, that kept the airflow from going that way. So it was an infection of 4 guinea pigs, and N was always equal to 4 here, which you could criticize, I would, as a very small number of animals, but it's probably very expensive to do guinea pigs in the lab. Mice cost about $1 a day per mouse. guinea pigs probably much more so. To do a study with 400 guinea pigs is probably prohibitively expensive. Plus, the guy didn't have a grant for this, because who gets grants for reading 80-year-old papers and trying something? So it's always N of 4. And then they look at relative humidity in the cage and temperature. And so here are the data from those papers. And what they're looking at is viral titers based on, does this guinea pig shed any virus at day X? And so they looked at it for 12 days. The dotted lines are guinea pigs they infected by sticking a swab in their nose and rubbing viruses. So all the guinea pigs who were deliberately infected produced virus and then either cleared it or died within a couple of days. I read the paper, they don't say whether the guinea pigs died or not. I don't know whether they get flu and move on. And then so at 20% humidity, 20 degrees, you see that in this case 100% of the guinea pigs shed virus over the course of the study. So 100% transmission, and then they looked at, say, did another trial at 20%, 20 degrees, 3 of the 4. And then no transmission at high relative humidity. So the conclusion being humidity affects the stability of the viral particle, which coincides with a lot of the epidemiology of the disease. Flu is almost unknown in tropical regions. So their hypothesis was that relative humidity mattered. Indeed, it does. Temperature, likewise, 100% transmission cold. I don't know if you can see this or not, it's kind of blurry. 0% transmission 30. So low humidity, low temperature, the virus is more transmissible. So flu happens during the winter because it's cold and dry. They did some controls to show, I didn't show the figure, the guinea pig response to the flu was perfectly normal, independent of conditions. So it wasn't, at least by any measure they checked their immune system. Interestingly, they shed virus longer on average, the colder it was. So it's not only more irritable, that's a measure of stability. And then they have a model for humidity versus temperature, and there's actually a little poem. This is speculation because they're talking about 2 trials of 4 guinea pigs each. But there you go, flu happens in the winter because it's cold and dry. We'll talk for just anti-flu or antiviral treatments. Of course, flu being such a huge virus, there are some drugs on the market. Most recently, Tamiflu and Relenza. They are neuraminidase inhibitors. So they're going to inhibit not the viral replication and exocytosis, but the release of that ligand. When you saw the scissors cutting the virus, that's what it inhibits. So it doesn't prevent infection, it prevents transmission of the infection from cell to cell and then person to person. Interestingly, this is getting a little into the weeds here, it's a rationally designed drug. It had chemists and computational people looking at crystal structures of the enzymes and saying, yeah, I can make a molecule that's going to fit into that active site. That's kind of an old drug. I mean, it seems like we ought to have better antivirotics now. There are several, one of which I looked into. Not yet FDA approved. There's one, Paramivir. It had an emergency use authorization during a flu outbreak a couple of years ago. It's a transition state analog. So inhibitors fit into the active site. Transition state analogs look like the high free energy species during cleavage. And so they fit into the active site more tightly than the substrate, neurominic acid, or the products. So it's in theory, if you're right about it being a transition state, a much better drug. So we see things coming down the pipeline based upon chemistry and binding and kinetics that tend to do a better job. And this is from a paper where they're looking at survival based upon a mix of Paramivir and an enzyme that inhibits the RNA polymerase. And so used in combination, they get a very good effect. So it's hopeful that we'll have some better antivirotics. See you on Wednesday.